President, members, and guests of Royal Society. It's an absolute privilege to be standing here and discussing what's an interesting topic, but also as the motto of RST stands, advancement of knowledge. I hope I can provide the advanced medical knowledge in this area today and how it has evolved into something that is being practiced at a number of places. Personalized medicine, it's also interchangeably used as a precision medicine or individualized medicine or standardized medicine. So these are the names that you will hear quite often from now on. In short, what we would love to discuss today is generally about the human genome and the status of human genetics before we had the phenomenon or one of the most important aspects in human science discovery called as human genome. And then how it transformed into genetic medicine. And I want to actually provide you with some examples of how it is actually is in practice. An exciting area called as pharmacogenomics, and then my own research area, microbiome and its applications, and how we are going to see this future of personalized medicine is the overall plan. So if you really want to define this particular term, personalized medicine, it is basically about the right treatment, at the right dose, for the right patient, and then we think of it as a right outcome also. So everything has to fall in the right place if you want to be precise with respect to medicine, and that's what we are going to define as personalized medicine or precision medicine. So before we really get into the deep genomic medicine, let's remember at the same time as the Royal Society was formed around the 1840s, 1850s, there was this Austrian monk, Gregor Mendel. I'm sure many of you remember him. He's called the father of genetics. Okay, so Gregor Mendel here, he was the initial person who actually proposed. Look, he used peas, okay, just the pea plant, and then showed a term called heredity. How are we getting these traits? Why is my hair dark and somebody else's is not? Or how is the skin color different, eye color different? He was the one who proposed all these into a term called as heredity. So, and we call the entire branch of his discovery as Mendelian Genetics. So for a long time, he was not recognized. Only in the 1900s, people realized, oh, wow, whatever this guy said was really good. He showed all that using these pea plants. So then there was a big pause until we were not sure about what is this inheritance material that is within our cells. As humans, we got around 3 trillion cells within the body. If I just break myself down to a single cell level, then what is the material? And that was in 1953 when Watson and Crick. So these are Watson and Crick in 1953 discovered the DNA structure. So that's when we heard the term deoxyribonucleic acid or DNA is the one that is giving us the genetic inheritance. So we get half from the mother, half from the father, and that is what is exhibited in what we are. Okay, so that was this one of the remarkable discoveries in human history called as DNA structure elaboration. So that is Watson and Crick in 1953. Then there was an expansion of wonderful human discovery around the DNA structure. Because we understood the DNA structure, we've learned a lot more about how we are, how we are able to move, how we are able to digest. All those things came into light, and that way we moved on to, sorry,
One sec. Sorry, just a pause. A couple of difficulties today.
So anyway, let me keep talking about this DNA. So then we know this is a fundamental structure that is within each of the cells and lives within a structure called as nucleus. Okay, so this is what it is. And the functional units for the humans are called as proteins. This is what we need. As I stand, number of proteins are produced. Even if I have to lift my hand, there are proteins that are working in the muscle. Okay, so these are the functional units of it. But we realized to make such proteins, DNA is the mother. This is where everything is coming from. And so remarkable, the whole thing, if I take out the DNA and stretch it, it can go to the moon and come back. It is that much. And how can it be, whether you call it as nature or God, whatever it is, the packaging is the best in the world. Imagine that is all packaged inside these chromosomes, 23 pairs of chromosomes. Okay, so why DNA discovery was excellent is because that showed this is what the material or the blueprint for the humans is this. And so we moved on then, yeah, it's okay. So then they showed, look, end of the day, it just looks like this. It's just a simple staircase. Hey, it's called as double helix. And you might see everywhere. But then if you break down the double helix, and then I say, what do you see there? We are nothing but a combination of 4 letters. That's it. That's what humans are. A, C, T, and G. So if I focus a little bit more here, this is the ladder. And then what you see is A and T. They combine only with that, and C and G. So in a particular combination, permutation combination of A, T, C, G, adenine, thymine, guanine, and cytosine, this is all we are. These are the 4 fundamental pillars, 4 letters of human life. That's it. This is what is built, packaged inside the cell. Supposing I want to make a gene of protein, what happens is it basically hones in onto that particular region, unwinds the whole thing. Unwinds it, that's what we call it as gene. So say I want to make insulin, only that insulin will be unwound. And then it makes the insulin protein. So this is what it is. So that's why the discovery by Watson and Crick was so amazing. We could explain everything about how we live based on the human DNA, how the inheritance happens. So this is what the principle of it. Then if you think about how genetically similar we are. So people looked at the DNA material and then said, how are we similar? The people in this room, we are 99.9 genetically similar to each other. So all that variation in your color to the way you metabolize, everything is just that 0.1 is the difference. And then how are we about chimps, 98.8% similar to a chimpanzee, but then we're almost 60% banana. So the genome or the overall material within each of the cells contained within 23 pairs of chromosomes is all this. That's it. That is a genetic material that determines everything within us. Okay? And then this is what I wanted to expand on this. So if you take a cell, this is a cell. Inside the central place called as nucleus, and that is what we are going to call a genome, means all the chromosomes put together, the 23 pairs. Okay, so this is what the chromosomes are. And within these, the DNA is packaged within those. Okay, so here are the chromosomes, they look like that. But then if we expand and look at the DNA and the gene, this is what it is, A, C, T, G combinations, gene after gene. And then, as I said, the fundamental units that do the job for us are the proteins. All those enzymes to chemicals, everything is, whatever is needed for human life is considered as protein. So you need that to make the DNA, and then in turn DNA determines what proteins are made. Okay, so that's what we are with respect to that. In 2003, as a scientist, one of the milestone things happened in the US where, and also other countries, in an effort from so many countries, there was a cracking of what is called as human genome draft. That means if I take a cell and inside, can you open and tell me how many letters are there? That's what it is. So they actually cracked that. They found out the entire sequence of letters, and then that is what we combinedly called as human genome. And you can see the New York Times editorial where it said genetic code of human life is cracked by scientists. It was a 12-year effort, and then it initially cost around $1.5 billion to get this. But that's a whole thing. We are sequenced. This is what we call sequencing. Each of those letters are known for humans. But still that we are similar by 99.9, and then they found within those letters, there's a tiny fraction, 0.1%. That's what we are different from each other. That's it. So that's what the discovery was, and then top journals such as Nature and the Human discovered it was a phenomenal discovery and well covered by media and so on. But this opened the field so beautifully. Now, earlier we were not sure about the structure of the human genome. Now we got the full package of human genome described in detail. Okay? That means we now can figure out where the problems are within those letters. Okay? After all, the genetic problems, everything lie in that sequence. Okay? So the 3 billion letters that form that is the human genome. So as I said, this is only 4 letters we have, A, C, and T, and G. Adenine, cytosine, thymine, and guanine combinations. That's it. That's what we are, the whole DNA is like that. But the whole genome, as I said, if you stretch the whole sort of sequence of letters, they are 3 billion base pairs. So the letters are 3 billion long. So 3 billion base pairs. But we talked about the gene, right? So the gene that opens up within the cell and that gives the proteins to us, there are only about 30,000 of them. So while we have a huge set of letters, only about 30,000 of them are what are called as genes or segments within that. So a short one. If we actually put it as a percentage, it constitutes about 1% of the genome. That's it. So only 1% out of the 3 billion is really important. That's what we realize. They are the ones called as genes. In turn, they make the proteins. What about the rest? Are they junk? Initially, people thought they are junk. Now we realize they are not junk. They are important in assisting those genes. So there's nothing called junk. Previously, for a long time, even in the 80s, people thought of this junk DNA. Then we realized some regions are so important for susceptibility or helping us preventing diseases and so on. So they do play a big role. But the genes are only about 1% of the entire genome. Okay? So if you actually put this together, if you want to understand the genome, you take our normal Launceston phone book. I don't know, people don't even see these days, right? Hardly see the phone books. But remember, earlier in the 80s, Telstra used to give this book. So you consider that book. And if you consider that book, about 150,000 phone books pages are needed to fill this. Okay? So that's the sequence of our DNA, our letter, our genetic code. Okay? And so what we understand is a disorder or a disease can be caused by variation within those base pairs. Okay? So as I said, they combine as what is called as base pair, A and T, adenine and thymine, cytosine, guanine. This is a base pair. So if there's a problem, if there's a letter change, that's where we call it as variation. So you are different. Whereas my sequence could be ACG. Somebody else's could be ACC or CAG. That's a difference. And that is enough to cause problems. Okay? So we are going to call them as single nucleotide polymorphism. Polymorphism changes, multiple changes. SNPs, SNP is a common name. Or mutation. If it is a bigger one causing chunks to be removed, then you call them as mutations. They are changing in their order. So now, imagine the challenge, 3 billion letters, and within that I need to find that. Out of the 3 billion, I need to find, there may be only one change we need to find. So earlier it was described almost like a needle in a haystack sort of arrangement. Very difficult to figure, but if you put the algorithm, it is worse than a needle in a haystack actually, because you can find the needle if you apply the algorithm. So it was an enormous challenge. And the cost of it, initially about $1 billion to get the first copy, more than that, but even if you keep it as $1 billion, and how we have come today in 2022 actually, now it is, some companies can do it for about $600. I think it is rapidly coming down to $200. Imagine 2003 when the first copy was released, $1 billion it took to just sequence it. And the rapid advancement following that has resulted in what we are going to soon have, a $200 genome. So you can send your sample, and then you get it probably about a week's time. I mean, at labs we do it faster. Within a day we can get the sequence. So $200 has come from 1 billion to this today. With the now enormous advancements in a number of areas. Now this is what is an example of a single gene Mendelian disorder. So if I wade through the entire sequence, So this is the letters I was talking about, 3 billion of them. And then this is the region for cystic fibrosis. Within a particular chromosome, I have to hone in on to a particular region and say, oh, this is where the difference is. So CFTR is the name of the gene, okay? And people with mutation, they cannot exchange salt and water. What happens, the mucus becomes so thick. So it occupies the lungs and gut, they can't breathe. So cystic fibrosis, the research was going on, but what has happened is because of the genome copy that was available, we are able to type a number of people, mild disease to, you can classify them under 6 headings. And then now we are able to pinpoint the mutations where the sort of polymorphisms happen, but the gene is, that region is the gene. Okay, so we are able to work out multiple things because of that. So if you take such Mendelian disorders, what we mean by Mendelian disease is single gene. That's all, only one gene is involved, and within that there's a simple region, and we can easily find that out using these advances in genome sequencing. So by the time genome sequence was available, we had about 1,700 Mendelian diseases discovered, but that time I was part of some diseases also. At that stage, it was extremely time consuming and so long it took. It was only by sort of approximation. Okay, this might be that. And so we took so much time for the geneticists to figure out all this. But once we had the genome available, about 5,000 diseases have been discovered. So some of these would have been missed. People who were diagnosed earlier in the 1990s even, we wouldn't have known because we didn't have the facility. Now the whole genome is available. You can just scan through and figure out all these things. Then this is a big deal, linkage mapping in families. Then what scientists did is to assemble families after families and say, can we find the linkages using the sequence? Yes, we could. And so lots of additional diseases have been discovered because of those things, because of the direct genome sequencing that was available. So if you look at the graph, this is the status. And then as we now move, Mendelian diseases have almost all been discovered now. So easy to look through the genome and figure out if it is a single gene problem using families. So this is another success. Now if you look at what are the common diseases, so because the genetic architecture is so clear, now we can work out even polygenic diseases. This means it is not a single gene, but the problem arises from a combination of genes. Okay, single we defined as Mendelian, but polygenic means multiple segments, multiple chromosomes. We have 23 pairs, right? So out of the 23, one problem could be in 6, the other one be in 11. So we are able to find that out now because of the genome sequence that was available. So we call such diseases as highly polygenic. Okay, so we are able to map 100,000 loci for hundreds of traits and diseases using the genome sequence. So it may be anywhere, multiple chromosomes, because we have the letters available to us completely, we're able to figure that out. Okay, so these are all the advantages of this genome, and it has yielded a field called as genomic medicine. So we are now able to figure out biological pathways to multiple diseases. For example, schizophrenia is not a single disease, it's a polygenic disease, it's a complex disease. And one of the diseases I study in detail, we are part of this, lots of discoveries in genetics, is called as Inflammatory Bowel Disease, IBD, Crohn's disease and colitis. What's interesting there is, there is about 225 genetic loci. That means, imagine CFTR or cystic fibrosis, one gene. That's all, we could easily figure out it's only in one gene. Here it is 225 genes contribute to the disease. Okay, 225, it's more actually now. So that many genes are predisposing people to inflammatory bowel disease. So this is the most complex disease known to humans. But even there we have made tremendous advances by working out the genome sequence where the trouble spots are. All those were possible. Alzheimer's or heart disease, for example, there are 95 loci that are important. And what we figured out is 80% of those do not affect the lipid levels. Initially, we thought the good guys and the bad guys, and we now believe that HDL is not protective, but the triglycerides confer risk. So lots of well-known things in medicine have to be rewritten now because we are able to precisely pinpoint These are the genes involved, these are the pathways involved. Is it absolutely accurate? I don't think so, because any condition, complex condition, involves genes to a certain degree. Yes, you are predisposed, but then there has to be what is called as a multi-hit hypothesis. That means there is the diet, there is your environment, all these things clicking, then you get the disease. So it's not only these, but we have understood at least this part. Many a times, for example, in IBD, inflammatory bowel disease, we know this constitutes about 20% of the disease, the genetics part. So at least we are able to crack that. So you understand that these are the genetically predisposed individuals. So I could sequence it and say, look, you have a 24-fold chance of getting Alzheimer's by the time you turn 55. That much precision we can do now. But the algorithm still has to involve a number of other factors, right? So, which we'll discuss a little bit later as well. So these are the things. And then we understood what are called as common and rare genetic variants. So there are certain people show very rare variants, variants meaning changes in the genes. So we are able to find the 100,000 associations in hundreds of human diseases have been cataloged now. So we have a huge catalog or a library of all these changes that are available. What would prevent that from coming to GP's clinic is the power of the computer. That's the lacuna at the moment as far as my research goes into all these things. We need supercomputers to work this out at the moment. But as the research is advancing, there will be a day where, my prediction is about 5 years, where we get this done and the GPs will have access to this. So your whole sequence, everything will be available, your predisposition will be available to a number of diseases. So it is prevention as well as treatment, both can happen through genomic medicine. So earlier, so if you take a common variant and then the genotyping, in 1998 it was so hard, one at a time. These days, we are able to sequence 1 million sequences in a matter of hours. In fact, there is last year, a company released a handheld DNA sequencer. Okay, so you just drop blood, just like our doctors, how they do the test for other things, sugar and so on. You can do that, and then that is able to sequence within about 2 hours. So we have come to that level. So that is why it is exciting to be living in this, where we are going to see these advancements helping us into genomic medicine, all these translating into genomic medicine. Then there is Genome-Wide association studies. So many countries, what they've done, they assembled families after families of disease groups, and then say, look, can we study you through what is called as Genome-Wide association? And that has yielded a lot of good things where we are able to pinpoint which pathway is missing within you. There is certain chemicals that are missing in this group of people to that level. So precisely we are able to identify a lot of things. The scientific signature within each of those diseases can be identified by GWAS or Genome-Wide Association studies. So what are the sort of powerful driving forces with genomic medicine? I put them under opportunities/challenges. Human cohorts are important for us. So you can now people are, hundreds and thousands are available. So we are thinking of hundreds of millions of people. So we can catalog all those things. So we can identify the common variants, but also rare within a population. So in this case, the precision medicine for the whole community will be available soon. So we are able to say, in Tasmania, there is a particular group that has got a particular mutation that is predisposing them to dementia, something like that. So in fact, that is what Menzies dementia group is doing. So they will utilize the genomics also in their studies to augment their studies. And then the powerful genomic technologies, as I explained, the sequencing technology. And then we are also creating what is called as a human cell atlas. So that means a single cell analysis is made much more easier now with the technologies. The third aspect of this is the data science. I think this is where the problem is. The cloud-based platforms and the security issues surrounding all those is a problem. And Eric also alluded to insurance and all that, but those I think will be sorted out. But the data source or the power of computers available at the individual GP level is a big problem now. That is where these are not reaching. Whereas the sequencing is not a problem. You can actually send your individual blood sample in the US, or there are many other places where you can send and get sequenced. But the interpretation and then how you link that to all your problems in consultation with your GP is going to be a problem without the availability of supercomputers or the computing power. So that area is a little bit of a problem at the moment. But otherwise, genomic medicine has taken up beautifully, and that is going to be exciting for the medical profession to utilize. So just a quick example as to, at least in the field of cancer, the precision medicine has yielded excellent results. And so, for example, what they do is, if you take a tumor, you can understand the diversity and heterogeneity. You may call a cancer as a breast cancer or colorectal cancer. And I can tell you with our own research, about 90% of the people have different variations within that cancer. So you call it as colorectal cancer, but it is not. Individually, we call it as a buildup of mutations in cancer. Okay? So cancers don't arise just like that. They take a bit of time. As I said, multiple insults from your genetics to environment has to play a role to develop it. So it takes a long time, and then we call them as building up of mutations. So number of genes are involved. So we are able to figure that out from the tumor level also. You can take a sample and put it through the genomic sequence and say, these are the mutations that is specific to you. Whereas person B, it is different. Right? So that's what we are saying about genomic medicine. It's going to be so individualized because I can figure out your data with respect to the genetic changes. Okay? Then the tracking the evolution of individual cancers is also possible because of that. Okay? So even for drug-resistant phenotypes could be identified if I do those. Okay? And the third aspect is the tumor microenvironment. For example, the blood vessels, because they're after all scavengers. What they do is they pinch everything else, the surrounding tissue. So we are able to now work out the microenvironment in which a cancer is placed, and we can work that out. And finally, the drug resistance mechanisms can be worked out. Because all thanks to genomic medicine, we are able to figure that out and say, look, this is the individual cancer problem that exists within this person. And so this is all the success story we got at the moment, from chemotherapy to hormone therapy, and wonderful drugs have arrived, as you can see the evolution. And one of the best stories I just want to describe a little bit more is called as the ABS, NIBs, and NABs. Have you heard of this? These are monoclonal antibodies, and then the small molecule inhibitors. Now, we need certain drugs that can only attack the cancer cells, not the surrounding cells. Now, can we do that? Yes, we can, because we are able to know through genomic medicine what is a cancer cell expressing. And we can actually make the antibody only to kill that, or present only on the cancer cell. So that is the advance, and this is all the monoclonal antibodies that are available now. And what used to be a death sentence in the form of blood cancers is no longer, it is absolutely curable. A number of blood cancers are curable because of these advances. In fact, the global market is about 60 billion worth with respect to the monoclonal antibodies. So you can see how expanding, and this is the good news story of genomic medicine, if you ask me, because we are able to precisely diagnose the cancers, number one. Number two, individualize the mutation responses. Number three, these cancer antibodies are available, and we can tell you, oh, look, this is not for you, because I know the sequence, it's not going to work for you, whereas it will work for somebody else. Okay, so we are able to classify, stratify cancers and individualize therapy. These monoclonal antibodies are really, really changing, life-changing drugs that are available. I mean, acute lymphoid leukemia, acute myeloid leukemia, those things were death sentences. Not anymore. The people are getting cured completely. It is just eliminating the whole thing. So this is just such a lovely story out of this genomic medicine, the monoclonal antibodies. And then the NIBs and NABs, which are the small molecule inhibitors. And fascinating therapies using immunology, also called as CAR T cells, where we are able to program the cells to kill the cancer cells. Okay, so this is on the genomic medicine side. I just want to share some snippets of, to me, this is the most exciting part about personalized medicine, called as pharmacogenomics. As you can guess from it, what we are trying now is drugs and how they work individually, right? Previously, if you take the drugs, when you take any medicine for that, basically the medicine has to be absorbed. That means how will it get into your body is what is called as absorption. Then the second part is distribution. That means where is it actually going? I mean, many a times you take ibuprofen and thinking that it will cure my headache, but you're taking orally, right? It has to reach where the ache is and go. So that's what we call it as distribution. So first it is getting absorbed, then it is distributed, then the important part is called as metabolism. How is it broken down? And then finally excretion, because you don't want them to stay after they do the job, right? So it has to be excreted. So in short, we call the whole thing of pharmacokinetics. In other words, drugs, how do they work within our body through this sort of four-way admin, okay? But remember, there are again multiple variables that impact medication response. For example, dietary supplements, diet, adherence, concurrent prescriptions, environment you are living in, smoking status, route of administration, whether it is intravenous or peroral and so on, hepatic function, how is your liver? Because that's a center for drug metabolism. How is your liver function? All these things will impact. But so if you put that algorithm, so these are the variations within the human. So these are same dose of the same drug in 4 different people. Okay, look at this person 4. He is extremely toxic. Whereas this person, it's okay, it's working really well. So putting it all together, you can divide the activity of drug metabolizing enzymes. So we are going to the genes and proteins now. We are working out what are those pathways by which drug is metabolized and what are the players there. Okay? Then we work them out as metabolizing enzymes that are important in individuals. So with that way, if you take this as a normal metabolizer and give them a score, this is called as activity score. So this is actually happening in a number of clinics, Mayo clinics. I don't know how many of you heard about Mayo clinics in the US. They are the pioneers who have done this. So this is, what I'm talking about is what is happening every day there. Okay? So you can, on the left side is intermediate metabolizer, or a person who is intermediate, very poor metabolizer. You can take the same dose as the next person, but you are not metabolizing it. The drug is actually getting wasted. That's it. It's not going anywhere. You think it is happening, but it's not happening because of these problems. The metabolizing enzymes are varying amongst the individuals. And then we have rapid metabolizer. Oh, look, headache finished in 30 minutes. Or ultra rapid metabolizer, 10 minutes it's done, the job. So we are so different. Out of these 60 people living here, sitting here, each one can be classified into a bracket like this. For the same dose, same drug. So what they've done, it turns out about 12 genes are important for pharmacogenomics. That's it. It is all contained, whatever I said earlier, as the activity score, you can put them all into only about 12 of them, 12 genes, that's it. So most important ones. I mean, there may be rare ones for people also, but by and large, you can put them into the 12 of them. So these are the ones. These are the 12 genes that are important. Okay. And what you can also see here is if I add, whether it is 0, so permutation and combination of all these, you can describe like this. So about, for example, about this 28.8, say for about 30% of people have a problem in about 4 of those genes. So we could put that all, this is a study by Mayo Clinic using about 10,000 people. So it's a decent study to show that, hey, our drug metabolism is so different individually. We are not the same with respect to how we metabolize drugs. That includes absorption to excretion. At many pathways, we are different. So this is an interesting study where they looked at one of the genes and said this is a global distribution from different continents. And yeah, predominantly but there is also these groupings, intermediate or extreme groupings. Even up to 20% of people all over the world could be different. So these are then what they did, if you look at your medication and then the major gene that is problematic, where it has got mutations. So we assembled all this and then say, okay, if you take, say for example, warfarin, the major problem between individuals could be with what is called a CYP2C9 gene. Or if you're tacrolimus, CYP3A5 is a major one that is going to influence how this drug is metabolized. And similarly, you can go on for many of these drugs. So we are now able to individualize and say, if I sequence your 12 genes, which is not a problem at all, this is on a chip. We just take a sample, run it through the chip and say, it gives you a score for you. And say, look, you are not metabolizing the gene number 6, and hence it won't work for you. This is the alternative drug. So these are predominantly described as activity scores. So you're able to assign the activity score for each of those. Okay, so we are able to do such things, and hence the personalized drug principle comes into. So for example, if I take one size fits model on top, this is like the group of individuals here, and then the same treatment if you give, the efficacy and toxicity vary. Right? So if you can be in one of the 4. Whereas now we can do what is called as pharmacogenomic model. I can use the genomics to classify all those genes responsible for the drug metabolism. And then I can classify them into groups where it's going to work, or I need an alternative drug, or I need an alternate drug and a dose, or I just need an alternate normal drug and dose, or treat with alternative dose. So we are now able to be so precise with respect to the drug and how it's going to work, whether it is suited for you or not. Can I change the drug? So that saves a lot of money, effort, and it is also very precise to the patient. We know that it will work now because it is based on so much research for each of those genes. And this is the third part I want to talk about, which is my own area. So we have done a lot of research into microbiome, and how it can be utilized for precision medicine. So without going into too much of all those, I just want to show the latest data where it shows we are able to talk about diversity. End of the day, this is nothing different to our Tasmanian ecosystem within forests or a lake. It's exactly like that within that. What we are talking about is the microbial diversity within each of us. And based on a number of factors, it is going to be different. By the time a child becomes a toddler, the microbiome settles in. We're getting what is called as a stabilized microbiome, only at that stage. And then what happens based on your antibiotic consumption and so many environmental factors, it will vary. But then there is what is called as a common microbiome. So we are only measuring in terms of that diversity. For example, if we take antibiotics and kill all the microbiome willy-nilly, then what will happen is the bad guys who are waiting on the wings will take over that spot. So you do have what is called as a niche, and the niche is a combination of viruses, bacteria, bacteriophages, archaea, fungi. They all work in concert to help us. So the beneficial microbiome is only by the term of diversity. If you destroy the diversity, for example, completely on fish and chips for a long time, no fiber. And that will screw your lactobacillus, Clostridium. These are the groups, names of the groups, beneficial groups. If you do that constantly, then the bad guys will take their spot. And the important thing is it's a double whammy. Not only do they take the spot, but they also secrete bad things, even cancer-causing things. That's a problem. So that is why the diversity is important. All the therapy is aiming at getting the diversity back. How can I establish that ecosystem neatly back? So that's what we are talking about. And within that, we have figured out for each of those diseases, what are the beneficial ones within that. So you take in cirrhosis, these are the guys that are important. They go up, and you need to adjust that. In fact, now if you take your stool sample and send it to a Brisbane-based company called Microbiota, they do all this beautiful sequencing and compare all the bacterial genes, and then they tell you, look, you are lacking certain important ones, and that can be achieved by eating more broccoli and beetroot. No, I'm telling you, because we work with them, and that's the report they give you now. They even go to the level of your metabolism or by these bacteria. So you are feeding them with good things, such as fiber. They just love fiber. In turn, they give us so many good things in a big basket called as short chain fatty acids. There's many of them within that. What they do is they energize the gut and keep you so healthy. And we also know they play a central role in overall health. So to the extent that in medical books now you'll see microbiome described as an organ, just like liver and kidney, microbiome is an organ now. So this is what the map of diversity can be created like this by looking at each disease and say what is really missing in them in a big population of people like that, whether it is IBS or IBD. This is what we work with. I've got to show you quickly about a couple of examples of how this microbiome precision therapy is working. First example is our own area in inflammatory bowel disease. This is a small intestine. So if you take this small intestine, it's a single layer of epithelial cells. This is what it is. This is a normal gut, small intestine, okay? So your ileum, jejunum, and duodenum. This is how it looks. This is where the food absorption will happen. And generally what we have is 2 layers of mucus. So 2 mucus layers. And the second layer here is impermeable. Whereas the first layer is where all the beneficial, the bacteria, beneficial organisms will be placed. Within that, in Crohn's disease, what we figured out is there is a particular bacterium called as Faecalibacterium prasunitzi. I took a long time to pronounce it properly. Many talks. So F. pras is an important organism. Upon eating fiber, it produces butyrate. So this is the golden molecule they call these days. There are about 200 companies that are producing this now. So this golden molecule is only produced by this particular group of bacteria upon eating fiber. Supposing in Crohn's disease, this is what majority of patients have. They do not have this peacekeeper, okay, called as Faecalibacterium, nor they eat fiber. So this is a double whammy now again. They are not consuming fiber, and the bacterial level goes down, and then this is what you get, the disease. So this is an excellent specific example where you can supplement them with this particular bacterial group and then hoping that it will work. I mean, this is ongoing. It is not a completely proven thing, but this is a research that shows an example of microbiome and how it can be precisely applied. This is another excellent example, and this you will see more and more, Akkermansia. So overall, microbiome research shows this is the most important bacterial group called as Akkermansia muciniphilia. It lives in the mucous area and helps. So what they've done, I mean, this is a sort of big research study to cut it short and tell you the summary of this. Basically, in metabolic syndrome, people struggling with metabolic syndrome, multiple metabolic diseases. If you supply them with Akkermansia alone, that's enough. Not a combination like what you see in chemists, so many bacteria together in probiotics. But this is enough. We call this as mono-association. Just one bacterium supplemented to these people, they're able to reverse metabolic syndrome. So this is really breaking barriers with this particular organism. And very soon you'll start seeing this in chemists. It's already available in some countries, Akkermansia. And the third part, which is my favorite part, is fecal microbial transplant, otherwise poo eating. But don't worry, because these days we have capsules. When it started, it was really bad because you need to take somebody's poo, grind it in a mixie, and then put it in intrarectal, and then it came oral also. But it was, then people worked out how we can capsulize it. Now it is available as capsules. So these are the early days. Promising pill now, not so hard to swallow. So even in LGH, there was a wonderful case of what is called as Clostridium Difficile. Has anybody heard of this? This is, you can't treat with antibiotics. So people will have chronic diarrhea. They'll just visit the toilet 10, 12 times and they can't get out because it's just breaking the intestine. All it results is diarrhea, voluminous diarrhea. So a wonderful study. I mean, this is now well known, hundreds of studies now to show what we are showing in this particular case is what is called as a week 8 resolution with those people. And then what you find is an amazing, the first blue is the effects of fecal microbiota transplantation compared to conventional therapies. So we are talking about V8 restoration. 92% of people can be cured with fecal microbiota transplantation. So this to me is the number one best story with respect to microbiome. Well proven, most successful therapy using microbiome as a precision therapy. And I've seen one case in LGH where this person was about to die. He's now living because of this. FMT. Very simple now. The only thing is the donor identification. Because after all, you can also transfer a number of characteristics with the fecal microbiome. So you need to be careful. That's why usually relatives, a healthy relative is preferable. So a time will come where there will be a bank available for this. It's happening already in some places in the US where they create the fecal microbiome bank. So if you have conditions like this, they can just take one and give it to you. So, and finally, with respect to another important area is personalized nutrition. Especially as we all age, the problem, see, as you age, what happens? By 65, the microbiome completely transformed. Until then, it is stable. But suddenly, we have done a lot of work to show that actually, show that big drop in beneficial microbiome. And that goes with immune system. I mean, Katie would have spoken about it earlier. The immune cells, number of beneficial immune cells also drop. So what will happen? Until then, you might have proper bowel movements. But then you realize, oh, look, I'm getting into constipation. Lots of this. It's because the microbiome is changed. So a lot of research is needed in terms of personalized nutrition. Whatever I spoke before will be involved in this. For example, the phenotype, the microbiome, the metabolism, how you metabolize things, including food. You can work out now your exercise, dietary habits, and genetics put together will be the personalized nutrition. And so it is already happening. See, if you go, nutrition and dietetics are employing this, and there is an area called as nutrigenomics. Area completely utilizing the genomics into the nutrition area. This is another happening area, personalized nutrition. Okay. So if you think about this personalized nutrition, is this having an impact or not? Look at the FDA approvals. In about 2005, on the left extreme, we had about 5% of new molecular entities. 2016, 27, now it is close to 50% by FDA for personalized medicine approach. That means it's rapidly advancing towards reality. Whatever we talked about in genomic medicine, pharmacogenomics, personalized nutrition, they're all happening. And these are the 4 predictions that have been done by about 2019. They said genetics will be used in routine screening. Yes, we know that, especially cancer, it's making a lot of difference. You take this tumor sample, sequence it, and say, these are the types of mutations you have. This is the right drug for you. And then the genomics will become what is called as a routine care. Very soon, as I said, the only limitation is the computer power. Once we reach that, we'll be able to use it. So, just like how you get your pathology results, you'll also get your genomics results. And then the algorithm will automatically work out based on that. It will suggest to the doctor, these are the available drugs for you, based on what we find as mutational changes. The speed and accuracy of diagnosis for thousands of conditions, that is already happening. We now can diagnose lots of conditions, even prenatal, lot more than just Down syndrome and so on. Only about 3 conditions are diagnosed. Now we can find out 100 conditions prenatally because of genomics. And then the right treatment plan, like how we talked about the drugs. I think this is the area I think that has made a lot of progress. Using a panel of drug metabolizing enzymes, we're able to say, look, this is a drug, this is a dose for you. And so now you can even go one step and say, you need to take, of course, postprandial, that means after food, but also by 1 o'clock. Because why I say that is, it's interesting because the circadian rhythm, our sleep patterns also have an influence in this. They work that out as well using genomics. So the circadian is about half a dozen genes that control circadian rhythm, including the melatonin release and so on. So we are able to work that out. That's why I said the prescription future will be, this is the dose, also the time you need to take it. Okay, so these are all the sort of predictions, but I think about more than half of these predictions are already happening. So if you think about all these put together, I mean, you can't ignore the sort of psychological factors and the environmental factors. They're also important. I mean, that is where we are unable to work that out. It is extremely complex. Each of the cities are different. When somebody is in a crowded space, having pets or not, smoking status, everything is going to matter for this. So how do we put them all together into an algorithm is a big challenge. Whatever I said about genetics, as I said, only maybe 15%, 10%, depending on the condition. But what about the rest of it? 80% is environmental. So this is where we are now working out what is called as epigenetics. That means environmental influences and how they change a gene. They do. The gene changes based on environment is called as epigenetics. So now we have built what is called as epigenome. Then we say, look, condition A, this is Tasmania, a lot more oxygen is available, less crowded. What are the genes that will be impacted? By somebody smoking, or alcoholic usage, too much of it, or drug usage. So you put all these and build an algorithm for each of these. That will be useful. So it has to be a locale based, it has to be individual history based also. So all this put together is what I think will be the future of personalized medicine model, where we can train, we can do lots of evaluation, and then the patients will be stratified as treatment groups. This is already happening. So the Mayo clinics in US are prime examples of this. What they do is, when the patient comes, they actually do what is called as preemptive screening. So they do for normal people also, and then say, this is your map of drug metabolism. Okay? And then, or even when a patient is admitted, this is a routine. They have to do this, and then that goes to the doctor and say, look, don't give this particular drug. And this is the dose. You have to reduce the dose by half. That's enough. I mean, that goes for vaccine also. Katie Flanagan's research shows that. We are sometimes wasting so much of vaccine because exactly half of it is enough. Or we are overdoing things. So that is also happening. Precision vaccinology will work that out. But this precision medicine is not far to work that out. As I said, the two important challenges we have is how do you fit the environmental model there? Number two is computer power to do all this. So major points to take is the custom tailored medicine, and more preventive care will happen. The one thing I couldn't talk because of time is the utilization of gene therapy, utilization of stem cells, and then the 3D printing. This is all coming. This is also precision medicine, because I could take your own cells and grow the organ you need. That will be happening, and it is already happening for a number of things. If you consider Fiona Wood's work after Bali bombing, how beautifully those sorts of, and that has advanced now. Recently, Fiona talked about advancements within that. Individual patient cells grow and get the skin grafts done. So rejection is less. So that will happen also. And a lot more on the digitized medical information is the one that we need. And then improved safety with few preventable errors and deaths. I think that's what the doctors will appreciate. That's what is dear to them. They don't want the preventable errors to happen, and they don't want patients to die. So basically, this is sort of a final thing put together. Here I've added also what are called as Organon Chips. So for example, if you want to diagnose a certain thing, I can take a little piece of yours. We do it in our lab also. So what you can do is, I work with the LGH gastroenterologist, take a little biopsy material, and then with a certain cocktail of growth factors, I can grow a 3D model of that intestine. So all I need is a little piece of biopsy material. Anyway, they take it for diagnosis. So then I can work out a lot of things within that model. So this is what is called as Organon Chips. We can work out lots of diagnostic factors, and even the drug therapies that will work for them. Then there's a 3D printing model. Already heart, liver is printed by 3D printing. So with the appropriate sterile measures, this 3D printing will change the way we look at transplantation. You can print organs using that. So it is not research anymore. It is actually the patentable models have arrived for 3D printing. This is an exciting area, but it's already happening, right? Wearable devices, and that will increase. Wearable devices will go for a number of factors. Now we have, say, blood pressure, oxygen consumption, and so on, but we can increase it to a lot more of metabolism. In fact, I don't know how many of you know about the capsule endoscopy. So, right, so that itself is something really exciting. The doctor can remotely watch what's happening as the capsule travels. Sequencing, this is what we talked about a lot. This bioinformatics is no problem at all. We've got informatics power, but we don't have the computers to work it out at the level of a GP. Yes, big centers have it. St. Jude's in the US, they have this. They built a supercomputer. I think they spent $300 million or something like that. There, this is a routine for babies when they go. Genome sequencing is absolute routine. Pharmacogenomics is absolute routine. Because then they can put everything and say, this is the sort of a signature for this baby. Something like that. So that's coming. And then we have the 3D scaffolding and nanostructures are coming in. That will help. This is the area that we also work on T cells. And this stem cell area has taken off nicely as well. So this is another important area in precision medicine. So this is, I think, while I only talked about genomic medicine, pharmacogenomics, microbiome, and nutrition, the other areas are also there. That is helping in what is called as precision medicine area in 2030. So put together, if you ask me, hands on heart, are we there yet? No, unfortunately, it's coming soon in your GP, the new products, I think so. So thank you very much for kind listening. Happy to take questions.
Professor Eri, we have a room full of unique phenotypes here, and some of them want to ask you some general questions.
Definitely, thank you.
George.
Thank you very much, Professor. That was terrific. How do you see the insurance aspects of this turning out?
I think at some stage they have to accept it because that's a very good question. There are fora looking exclusively at this. Genomic medicine versus insurance, what will happen? So in the US at least it is a big talking point. They've made regulations already. Many insurance companies are including this. They include the premium a little bit more and say you can sequence now and provide it. Insurance won't be a problem, George. I think the main problem is the security. That's what we haven't worked out really well. Insurance won't be a problem at all because it is well known. Genomic medicine individually will be well known. Only, I know why you are asking this question is, if you say somebody is predisposed, will insurance take it? That is a very tough question to answer, I think. But patient, once they are admitted, using the genome sequence for disease stratification won't be a problem. I think that area is fine. But the other part, I'm not sure. It's anybody's guess.
Thanks very much. It was a very, very interesting talk. You said earlier on that the genes are stabilized when about the time somebody is a toddler. So what can you do, or what is done in the environment or whatever before somebody's a toddler that can affect the genes, and do we learn about that?
Yeah.
Do we know what to tell people now?
Yes, we do. So let me take a step back. When a baby is born, the microbiome of a baby is that of a cervical microbiota of the mother. That's what it is. When it is born, it resembles the cervix, the cervical microbiota. That is why cesarean section babies don't have that benefit. So what they do, in fact, we couldn't get the ethics for it. What they do is they take the swabs from the mother and then they put it everywhere, on the cheeks and the body of the baby. And that works. So let me go timeline wise. So that's how initially it starts. Then the microbiota is predominantly what is called as lactobacillus. Mother's milk is extremely important for microbiome settlement. So other research shows if the mother's milk is not provided, the dysbiosis we call, the ecosystem is changed. Those babies are much more prone to asthma and allergy. Because of the microbiome and the central part, it plays in disease association. So that is important. Even one step further, before this, if the mother smokes and consumes a lot of alcohol or drug usage while pregnant, is also dangerous. That has an influence as well. And then if they, as they grow, and the food variety, if we change from liquid diet to solid diet, and if you're not using the appropriate ones, that will change the microbiome. And then by the time they settle as a dynamic microbiome, that goes until about 65. That's the stage we call it as a dynamic microbiome settlement. That you can screw up with your diet and environment, everything. And antibiotic usage is a major factor in this. Constant antibiotic usage, it'll kill. They've done a study where they've given antibiotics and said, how long does it take for the microbiome to come back? It'll come back, but it'll take time. So it could vary from, for a person, 1 week to somebody 1.5 years, it didn't come back. So antibiotic is a main villain for all this. So by the time they settle down, the dynamic microbiome, and then however your diet to environment can affect it. So it goes through these changes all the time. But heavy C. difficile, for example, why is it happening? Because it's constant use of antibiotic. So at that stage, the bad ones have accumulated. In colorectal cancer, we know there are 2 species of E. coli that are responsible for secreting carcinogenic substances, the bad ones. And those are the people who are not consuming fiber at all. So the beneficial ones are slowly and steadily declining, giving way to the bad ones. So that is why the diversity is a major term and dysbiosis is a major term with respect to microbiome. So we can, the answer to the other question, precision microbiome will include that. So where we can say, can we supplement these people with certain bacteria? Right now it is not a success story. Hands on heart, I could say, you can have 100 probiotics. Do they all work? No. Because we don't know which is the right combination for you. The only one so far, the best one I found is Akkermansia. That is universally working for a lot of people. So that is why that is the best so far identified.
Yes, I've got a question about 3D printing to make a new organ. You would start off with a sample of the original organ and grow it from, is that like the source of the...
Yeah.
So you sort of grow it up and grow it up and grow it up, and then, but then how do you actually 3D print it?
No, these are 2 different things.
Oh.
So 3D printing can be just a liver straight away. So what you do is, you grow so we know the structure of liver, for example. Okay, so you know the hepatocytes and then how the veins and everything. So you produce those molds and also produce the right ingredient. The cells are supplied. They can be grown in the lab. That is, whereas the one I'm talking about you in terms of stem cells is, you can take a stem cell out of the gut, for example, and then I feed the right factors because the nutrition for the stem cells is important. So for a long time, we never worked out what they need. Now it is worked out. So I can supply with factor X, Y, and Z, and that grows into a lovely gut from your own stem cell.
Wonderful, thank you.
So these are the 2 different. 3D printing is different. Only thing at the moment is how do you keep it sterile? Because for transplantation, everything has to be sterile, infection free. So that's where they are working out. But 3D printing not a problem. Already they printed a heart. So you can do, because we know from anatomy, we know the structure, so we can put those into the printer and get them printed.
I'll let you go first. Excuse me, mate. Should a mother feed the baby from her breastfeed for 18 months or longer?
So what research, at least 12 months is needed, because as soon as the baby is born, nothing else other than the mother's milk can nourish and give the antibodies first. So you need the antibodies to protect the baby, right? This is what is called as passive immunization. Just like your vaccine, mother's vaccine is the mother's milk. It actually has got the antibodies ready-made, even better than a vaccine. In vaccine, you inject it and then you make the antibodies. Whereas mother's milk has got antibodies ready-made for the baby to survive. Number one. Number two, the nutrition supply is also given. So far, nobody in the world is able to make mother's milk. You can try so many combinations, salts, blah, 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 all those things. Never works. Only mother's milk is the most unique thing in the world. You can't replace it. You can't make it. So, number one is antibody and protection from infection and so on. Number two, the microbiome. Microbiome wants the mother's milk. As I said, more than 50% is lactobacillus. And they grow only on mother's milk. So to establish the initial microbiome and protect the baby, you need those aspects, and also infection-free, because they can fight infection only with mother's pre-formed antibodies. Mother provides it, ready-made. So the sacrifice of mother country starts there.
Thank you.
Now, what about an adopted child then? Because they're not breastfed at all. They're usually taken away from the mother straight away. They don't have any of those protections.
Absolutely. No, I agree. So the one aspect is, look, can we supply such kids, children with probiotics? That's what is happening. I mean, you can't do the antibodies side of things. But what you can do is prevent the infection side. Don't expose them to too much of infection. Second aspect is the lactobacillus. Now they are making baby probiotics. So to some extent we can give that, but I'm still suspicious about it. So they will be much more exposed to multiple infections later part of the life, asthma and allergy. These are the ones they'll be predisposed to. Apart from that, if you think about what is called as hygiene hypothesis, what we are saying for the human immune system to properly develop is, early stage of the life, they should be exposed to dirt. If you have bubble babies, they are not going to, they are prone to autoimmune disease, a lot more, and asthma later in life. So, but to answer your question, unfortunately, we can't do too much except some baby probiotics are available.
Are there any cost-benefit studies being done on, for example, the traditional versus new treatment of blood cancers?
Yes, so blood cancer is a major area of beneficiary out of genomic medicine to start with. So, for example, now what they do is you take out the cells, do the genomic sequencing, identify, and then what they had as, this is even retrospective study, people who are not cured, they compare, a lot of studies have compared that. Then they say, look, we are missing a certain pathway with a new genomic sequence we were able to identify, and that was the difference why it didn't work. So now we precisely identify, and even, for example, BRCA, one identified person, within that there are variations. Two people can be BRCA1 breast cancer, but then the pathways, accumulation of mutation is what we are talking about in cancer, and that could be 5 or it could be 8 mutations. So BRCA with 5 mutations, BRCA1 with 8 mutations. So this person needs a different drug, although the main mutation is BRCA1, but the sub-mutations can be identified. Similarly, blood cancer, what they've done is identified certain expressions. So on the surface of a cancer cell, they identify certain things that are precisely expressed in people with certain cancer. Now we target them with antibodies. So normal cells have nothing to do with it. So exclusively on the cells, and that is what is monoclonal antibodies. Huge success.
So would you see the new drugs being the first line of treatment generally, or only when the first conventional ones fail?
No, it is, once again, I think once again, one size fits all is thing of the past. You need to be individual. And already LGH hematology is doing that. Basically, you need to type the earlier we were doing, here is a person who has got ALL, acute lymphoid leukemia. And here is tacrolimus or whatever is a drug therapy, chemotherapy. Now they don't do that. They basically understand multiple parts of a cancer. Cancer cell is only one thing, but then the blood supply, and then what sort of nutrition it is preventing the normal cells from. And then they also look at the oxygen levels. So whole thing is considered, and then there is a package that comes to you and say, this is a drug that will work for all things considered. So that's the way, so that's how it is going to be for therapy.
Can this be applied to autoimmune diseases?
One area, I must confess, this is an area we haven't cracked anything, autoimmune disease. The interesting thing about autoimmune disease, here is an antibody that comes and attacks your joint or your particular myelin, even in MS. But what we haven't figured out is how to prevent that antibody. And so, unfortunately, autoimmune disease area, while genomics has yielded a lot of data with genetics, but in terms of therapy, we haven't done much in autoimmune disease.
I don't know if it's related to this, but I understand that gene therapy has been used to cure certain people of haemophilia, hasn't it?
Correct.
Which is great news.
So, very simple Mendelian disorders, you can use gene therapy. For example, Lorenzo's oil. How many of you have seen the movie Lorenzo's oil? There it is a particular amino acid gene mutation. You can supplement with a certain thing that is a source of that amino acid. But complex diseases, which are most of our diseases, very difficult to treat. All these autoimmune diseases fall into that category. Multiple genes, how do you work that out if it's not a single thing? So it's going to be problematic there for gene therapy. But I tell you the most exciting advance these days, it's called as CRISPR. Have you heard of it? It's a Nobel Prize recently for CRISPR. What they can do is, we talked about the gene, right? Can we edit it? Can I change the letter T to A? Why not, isn't it? I mean, that's a very fascinating prospect. I have a problem because instead of A, I have a T. That is causing my genetic problem here, right? Can we edit even one letter or multiple letters in various genes? Yes, you can. And so that ethical aspect is preventing at the moment. There was an illegal case in China where a person has treated an HIV patient's kid, and that was completely taken off after that. That's the world's first case of CRISPR editing. Means you can actually insert a technique where you can change a letter. Out of the 3 billion, go and change one. So you could do that, but that is, once again, ethically, I don't know. These are the complex issues with ethics, where you can, then we'll have a CRISPR baby separately. It's going to be a problematic area in terms of ethics. This is the thing, insurance and ethics are the two, and security. These are the three aspects that we have to consider in all this.
My question might be a bit too controversial at the moment, but I would love to know what you think about giving babies and young children the vaccine for COVID.
Yeah, the Australian government has recently approved a new vaccine that is for up to 5 years of babies. So when we do a lot of other vaccines, I don't think COVID will be a problematic one either, because now we have understood COVID more and more because of research, rapid research. Have you ever thought, what is the duration for a vaccine to be released? If you ask Ian Fraser, I used to work right next to him, okay? Ian Fraser's vaccine took 23 years from discovery to be the product that is given to our girls and boys in various schools, right? Whereas this FDA approved 11-month approval for vaccines. Never in the history has happened. So, and to go with that is the research work that shows how vaccines work. So, many people have turned into COVID researchers. Labs are converted from other areas. So a lot of research is happening. The knowledge is built up, and by reading, it shows babies can be safely treated with vaccine. So immunized. So that's going to happen. And already the Atagi, where Katie Flanagan is also a member of, they recently approved one.
You've mentioned CRISPR as a preventive, okay, which is quite controversial. So would it come to a point where you have individualized, everyone gets the genome sequenced, and from the start, then we could be talking about individualized diets in the preventive mode, or lifestyles. Okay, will it go that way?
Yeah, absolutely. So I can see, that's why I put a figure as 2030 is probably the time by then Launceston will have it. Because the challenge now is the environmental side of things. Each of the environment is so different. For example, sun exposure. We are not the same as Gold Coast, right? The skin cancer capital of the world versus, fantastic environmental, hardly much of UV effects. And so similarly, the smoking status of people and a number of other factors will be considered. And so the algorithm to be developed needs a computer power. That is what is lacking now. Individual research is available. For example, somebody who has got pets or less exposed to IBD, or they don't show IBD for a long time. Or many other diseases show that having pet is so beneficial. So similarly, living in a rural atmosphere is much better than an urban atmosphere for development of disease. So these are all examples of environmental effects. But how do you put it all together for a GP in New York to a GP in Launceston is a question. Whereas the sequence is the same, whether you are in New York or in Launceston, right? The environmental factor is so different. And individual history is so, I illustrated how a baby could be exposed to or predisposed to asthma by the mother smoking or by the mother not feeding. So when a 40-year-old goes to Dr. Preeti, how will she know that as a baby you are not getting the mother's milk? So history to environment, all these factors put together will be individualized medicine. Because we will be missing certain factors within this. So that's why all these, that will come where they say these are all the 18 factors that you need to develop this disease. Something like that will be available to the GPs at some stage. Genome is one side of things. I mean, that is not be all, end all. For Mendelian diseases, yes, if it is a single gene, celiac disease, or about 5,000 diseases are known to be called Mendelian diseases, single gene diseases, no problem, we can treat them. Even there it is a problem in the sense, some conditions, even cystic fibrosis itself, we got drugs now that can break the mucus, so it is not blocking the lungs or the guts. But even within that, we have identified people who do not respond to anything, even within that. So it's not going to be that easy, but it will change the way we treat because the doctor has got the genome available, microbiome available, nutrition advice available, and then at least it will be much better to treat. And the precise drug is also available to you because you are a metabolizer who is rapidly doing this, or you are not metabolizing certain things. So I think, yeah, my target is 2030 for Launceston.
One last question.
Thank you very much for a very interesting day. I think mine's more of a statement for you rather than a question, but all of this wonderful stuff that has been developing over time and up to where you've come to and the future, I still see that one of the biggest blocks we have, apart from security and all of those sorts of things, is individual attitudes, belief, religious beliefs, cultural beliefs, and just damn bloody mindedness that we have plenty of information that provides knowledge to people, but the exchange of knowledge to practice change is the huge leap.
Absolutely.
Hence, we still have new smokers and we still have sunbakers and we still have lovely red wine and red meat and so on, and I don't see that will stop. So this is for an exclusive part of society, I see.
No, this is not actually, because what they've done, for example, in pharmacogenomics, very good question actually. What they've done is they've done the Caucasian Americans and then the Americans of African heritage. And now, you're right, actually it varies. And so, that is also part of the personalized medicine because now you are treated as an individual rather than a particular race. And say, for example, in genetics, it is well known the Jewish community is a lot more predisposed to a lot more diseases. And that amounts to, evolutionary aspects are there. The reasons are explained through inbreeding and so on. But nevertheless, the genome sequence will identify that, irrespective. So that won't be a problem in terms of your belief or anything. So that is why the personalized genomic sequence will help you tailored to your genome only. So I can pinpoint, that is why I said if you see a typical report, you can say that they actually show you the fold probability also. You are going to get Alzheimer's by the time you turn 50. At least the genes are showing that this is what, because now we can predict putting all these environmental factors approximately, your chances are high for this, like that. So we can do that. So basically, genome will override the cultural aspects.
Well, thank you for Professor
Sorry,
Eri.
sorry, do you mind if I've just got one question. It's a little left of centre. It doesn't have a lot to do with biome rather, the biome itself. Epigenetics, there's some research currently done, very positive results by Professor David Sinclair in the US. Do you mind if I ask a question regarding that?
Yeah, sure.
Great. So he describes the lifespan of a person and the health span.
Yeah.
Putting environmental pressures on the epigenetics, either extreme high temperatures or low temperatures, and I suppose kilojoule deficiency has shown to extend the length of the health span.
Yes.
Is there any implications regarding that and how far you can sort of push the genetic treatment of a person?
No, it's a very good. David Sinclair is we should be very proud of this Australian scientist who has done so much good work. When you talked about red wine, it's because he's the one who established red wine has got good qualities. So it's a very good question. I mean, I've read most of his papers, and in fact, we hosted him in Hobart a few years ago. So the point there is what we haven't talked too much about is the chromosome itself, because the length of chromosome also has a play in longevity. So that's coming in. Now you can measure it and say, the lifespan versus the environmental effect on that one. The epigenome, again, is a big problem because individually we can do research in the lab and say, epigenetics is affected by smoking alone, or, the other environmental factors such as alcohol or sun exposure. Those are the things that individually you can study. How can you put all this together into a research study is a very, very difficult design to do. So at the moment, that is a problem I see as. So I can't say with certainty answer to your question, but I think in the future, once we work out how to model this using the environmental factors, we'll gain big time. At the moment, the environmental factors, which contribute to the major part of somebody's disease progression, is being worked out at the moment. Genome side, no problem. We have all the resources available to sequence and get that.
Wonderful, thank you. Thank you, Raj.
Thank you.